So welcome back. So this is the last session of our genetic introduction. So in this we are going to discuss what are genes, what are alleles, etc. So here as shown in the diagram, genes are nothing but they are portions of, or they are certain regions of the DNA, and these are the regions which are responsible for the production of proteins, or these are the regions which will be transcribed. So when you say that the genes are seen in certain locations, there are other regions other than the genes. Okay, and the regions other than genes, usually they are called as junk DNA. Junk in the sense that they do not have any or they do not code for any proteins. That means they are not genes. Only genes, or the segments of DNA which are called as genes, can produce proteins. That means they are transcribable into mRNA to produce protein. The other regions, they do not code for any protein. They are called as junk DNA, and the function of junk DNA is regulatory in function. They regulate the transcription process of the particular genes. So in this particular diagram which is shown here, you can clearly see genes as well as junk DNA. This is a portion which is shown in pink is called as a gene, and you can see the other regions which is shown in purple, that is junk DNA. So you can also, you might see another gene here, another gene, etc. So between the genes you can see portions which are called as junk DNA. So junk DNA is the DNA which does not code for any proteins, or it is not a gene which does not produce any mRNA to produce a protein. It is the region of the DNA which is having a regulatory function. Regulatory in the sense that it regulates the process of transcription. That means whether a gene is to be transcribed or not will be determined by the regulatory elements that is seen in the junk portion of the DNA. So that was the terminology which is called as genes. And coming to the other one, the other term which is called as alleles. So alleles are nothing but they are two forms of a gene. Two forms in the sense that uh, there is a gene for example, if I take hair color, there is a gene or a segment of a DNA which is coding for hair color. So you can call it the gene as a hair color gene. But every hair color is not the same. You can see hair color as black, you can see hair color as white, you can see hair color as brown, etc. So these different forms of a gene. So the gene is the hair color gene, and the alternate forms of a gene. Alternate form, for say you can take the skin color, you can take flower color. Flower color may be red, flower color may be white, etc. Flower color may be purple. The gene is, for say if it is the skin color gene, and the different forms of skin color are their alleles. So for most of the genes, in the case of diploid organism, we were talking about diploid organism. We are also diploid organisms. For diploid organism, most of the genes, they are having two alleles in the homologous chromosome. Mainly, they are having two alleles, or two alternate forms are present for most of the genes. But in some cases, there are more than two alleles, which we'll discuss as a separate section. But for most of the genes, you will be having two alleles or two alternate forms. Okay, so alternate forms of genes are called as alleles. So, for example, eye color is a gene, and there are different eye color. It can be black, it can be brown, it can be other shades of green, etc. So, the alternate forms of that particular eye color, black is one allele, then green is one allele of eye color, brown is one allele of eye color, etc. So, the alternate forms, or the different forms of a gene, is usually called as alleles. So, if for a or for a diploid organism, as shown in this particular diagram, usually they will have two alternate forms of a gene, or two alleles will be present. So, depending upon the alleles which are present, or it can be classified into, or it can be named accordingly as homologous, then recessive, etc. We'll come to that later. Next is another terminology which is called as locus. So these are homologous chromosome. So homologous chromosome we discussed in the earlier sessions. These are chromosomes with the same genes. As we said, this is R here, the same P is there. So they will have the same gene and the same gene order. The only difference is that they are derived from different parents. This is maternal homolog and this is the paternal homolog. So they will have the same genes in them, they will have the same gene order, etc., but they are of different different origin. One is paternal and the other one is maternal. That means one you get from the egg, the other you get from the sperm. That is maternal and paternal homolog. Next is locus. Locus is nothing but the position of a gene in the chromosome, where it is placed. So the place where the gene is seen is is called as the locus. So in a homologous chromosome, a particular gene will have the same locus. That is why you can see R, the small r's at the same position, this P at the same position. So in a homologous chromosome, the alleles will have the same locus. Alleles will be present in the same locus. So these are the alleles and they are seen in the same locus. So that is also locus is that. So at at each genetic locus, an individual will have two alleles, one on each of the homologous chromosome. So here there are difference that is given here. And the other terminologies used are homozygous and heterozygous. So homozygous is nothing but if a gene is having same alleles at both the locus. Okay, if it is having the same allele, that means if the maternal and paternal, for say I am taking the color, I am taking the hair color black, okay, and I represent it as capital B. If father is having black color hair and mother is also having black color hair, both are the same. Okay, so you write it as capital B and capital B. Okay, if the alleles in an individual, if the alleles are same, okay, if these alleles are same, you call them as homologous. Sorry, no, homozygous, not homologous, homozygous. Okay, it can also be white, not every time black. So if this is the case, that means both the parents are having white color of hair. This is also said to be homozygous. If the alleles are same at the two locuses in the homologous chromosome, it is said to be homozygous. That is why you call it as homozygous here. Okay, and another thing is if one of the parent is having black color hair and the other parent is having white color, gene for white color. Okay, in such a case, this particular pair is said to be heterozygous alleles. So these two are homozygous alleles and this is heterozygous allele. So if a gene is having two different forms in an individual, that particular gene is said to be heterozygous. If both are same, both the alleles are same, they are called as homozygous. So these are the two terminologies. Next is dominant and recessive. So dominant is nothing but in this particular heterozygote, that means heterozygous, that means it is having the gene for black and it is also having the gene for white color hair. But if in this particular individual, even though it is having both these genes, he or she is having the hair color as black. So what is happening here? The white color hair is not shown or it is inhibited. The white color hair does not show up even though that individual is having a black or a white color hair color gene in him or her. It does not show up, it shows black. In such cases, that black is called as dominant and the one which is not showing up is called to be recessive. So that's with that terminology, dominant and recessive. The one which shows up is called as uh, is said to be in a heterozygous condition. The one allele which is showing up is called as dominant, and the other one is called as recessive. So these are with the terminologies. You can assign this dominant and recessive only when it is in the heterozygous condition. In the homozygous condition, both are the same, but there is no relationship between, or there is no dominant and recessive relationship in a homozygous condition. But when you come to a heterozygous condition, in the heterozygous, you have two different alleles. The one allele which shows up, so if you have two hair colors, black and white, instead of white, black is showing up. That particular black is said to be dominant, and the dominant one is usually represented by capital letters, and the one which does not show up in a heterozygous is called as recessive, and that is usually represented by small letters. So that is the difference. So always dominant one is for sake of our convenience or for our understanding, dominant and recessive. So dominant is the one which shows up, and that is usually designated by a capital letter. The other one is designated by a small letter. Okay. So now we'll take the example of mitosis with this meiosis with this particular thing. That means what will happen in a heterozygous one, or how it is being in homozygous and heterozygous. So I am taking, sorry, we'll take only the chromosomes. Okay, I am having a homologous chromosome. I am taking hair color. First, I am taking it as a homozygous condition. Both are black. It can be white, black or white, whatever it be. So now this particular one is going to undergo meiosis, and it is going to produce sperm or egg, depending upon whatever it is. So what will happen to this? So this is a particular cell. What is going to happen in meiosis 1 and meiosis 2? So in meiosis 1, what should happen? In meiosis 1, this is the homologous pair. They will separate and segregate. Okay. After meiosis 1, you will have two cells. In both the cells, you will have this capital B's. Is that clear? And in the next one, which is M2, as we said, mitosis, both the sides are attached. What you get is like this. So everything is capital B. So in such a case means, if you are having, if you are we'll be taking, we are considering only one character. This particular chromosome may have n number of genes. We are considering only the hair color gene, which is both black. Okay, if the hair color is or the particular genus in homozygous condition, the end result is it can be either sperm or the egg, which is produced here, 4 sperms or 4 eggs. The gametes which are formed are all same, right? Everything is capital B. In such a case, when you do genetic problems, so this should be kept in mind. If it is a homozygous, if you are taking a particular character, a particular trait, for example, a particular type of gene, which is hair color gene, and we are considering it as homozygous, the gametes produce. So we cannot write all these steps when you do a problem. So this should be kept in mind. If it is homozygous, if they are homozygous, they are producing a similar type, or all the gametes produced are of the same type. Everything is capital B. As a result, when you do genetic problem, when you write the type of gametes, okay, you cannot write all these 4. Only one type of gamete is formed. So if you are writing an individual as capital B, the gamete formed will be only one type, which is capital B. You don't no need, when you do genetic problem, please keep in mind that when you are having an individual with homozygous alleles, it will produce only one type of gamete. As a result, when you write gamete, you need to write only one, because all four are the same. It is capital B itself. As a result, you need to write only one gamete if you are having a homozygous individual. So we'll discuss that when you come to genetic problem, this is because we discussed meiosis, etc., homozygous, etc. So yeah, this should be kept in mind. That means if you are considering a character which is homozygous, that will produce only one type of gamete, and you need to write only one gamete in such a case when you do genetic problems. So just keep in mind this particular, take it as a rule. So this rule should be kept in mind. That means when you consider a character which is homozygous, all gametes will be the same. All gametes can be the same, and because they are same, you can write it as a single one.